And thank you, Bal Ganesh. It's a great pleasure and an honor to be here. As Bal Ganesh said, he and I go back a long way. And principally, my admiration for the approach he has taken is comes from his being founded, anchored in genetics in multiple ways, and that familiarity with genetics is something which I shall allude to in my talk. Um, Genetics has this beauty that you can infer what is going on inside the organism or inside the cell without ever looking inside it. And that power led to great understanding in many areas of our attempts to understand how life works. However, combined with such approaches, scientists also started looking inside cells and inside organisms. And that led to new kinds of revelations, most of which verified many of the approaches taken by genetics, but also opened up, as we see today, the enormous ability to get data from cells, from organisms, from tissues, from species, and do that over long and short timelines. This tsunami of data poses new challenges to us about how we can analyze both how life works and also to deal with the consequences for health, for agriculture, for society, and so on. So, in other words, the lack of availability of data and the requirement for thought and interpretation led to many advances. And the huge availability of data now should therefore lead to even greater advances and even greater conceptual understanding, but that poses a big challenge in how we analyze the data. So I've titled this talk Phage and the Origins of Molecular Biology, and when I wrote to Appaya, I said that I will talk about the origins of molecular biology, and I also said I'll talk a little bit about the work in my lab, but I think I'll leave that part, the latter part, out. I'll talk about a little broader other kinds of perspective. What I'll do is to tell you where this term came from, and it came from this book edited by John Keynes, Gunther Stent, and James Watson. And then I'll go on to tell you about, in the context of how, by looking at simple phage and bacterial genetics, we got a great understanding. How did our ability to get that understanding come about? And then how have we gone on to develop understanding in new areas? Many of the leaders of phage and bacterial genetics, when they had done their work, they had a hubris that they had solved critical problems in gene regulation and gene expression, and now they must solve new problems, and all of them went to study, most of them went to study the genetics of behavior or the nervous system and how it mates and how it functions. That challenge, that problem of solving how the brain works, is a problem which has not yet been solved, showing that extraordinary accomplishment in one area doesn't make it easy to get accomplishment or solve even more complex problems. So what is the complexity of the brain or other tissues which makes it recalcitrant to these kinds of solutions? Now, this book was edited by Keynes, Stent, and Watson, and it's a seminal work in the history of molecular biology. It was published in 1966 to celebrate the 60th birthday of Max Delbruck, who is illustrated here in a Picasso-like painting. And it's a collection of essays written by Max Delbruck, Salvador Luria, Alfred Hershey, and these were all central figures in the phage group. And the phage group really dominated our understanding of biology and gave rise, in due course, phage and molecular and bacterial genetics gave rise to the term molecular biology. And this term molecular biology was configured by a person called Warren Weaver, who was an administrator, top administrator in the Rockefeller Foundation. and became very popular. And when Erwin Chargaff heard of this term, he was upset. Chargaff used to get very easily upset, and he said that what other kind of biology is there? What nonsense is this? Chargaff was a biochemist, and to him the term molecular biology was redundant. All biology is, after all, molecular. So the book highlights how phage research provided crucial insights into fundamental biological processes. And for example, studies on phage led to the Hershey-Chase experiment, proving DNA is the genetic material. It captures the collaboration and the competitive spirit of early molecular biology at institutions like Caltech and Cold Spring Harbor. For example, Seymour Benzer's work led to the R2 region of the phage, led to a fine structure mapping of genes. So it was a very exciting period. So the book had enormous influence on molecular biology, and it underscored the importance of model organisms in understanding fundamental biology. No one at that time questioned too much that the regulatory mechanisms which people discovered in phage were not applicable to other organisms. They didn't say, oh, what you have discovered is there in phage, I don't know whether it will be the same in mouse or not. In fact, it was the other way around. People thought that all genes would be regulated the way phage genes are regulated, and phage and bacterial gene regulation. There were subsequent editions, and I would urge you to get hold of one of them. In 1992, there was another edition. So this is after 1966, and that edition looked at how molecular biology had evolved since its first publication. And there was another one in, I think, 2017, which summarizes the current situation. Now, the interplay between bacteriophages, bacterial genetics, and molecular biology has shaped our understanding, as I've said. And you know better than anyone else that bacteriophages gave a great understanding to gene structure and function. The experiments with phage lambda provided insights into DNA recombination, the intermediates of recombination, and all the regulatory mechanisms. And also, phage, as you know, facilitates gene transfer between bacteria and horizontal gene transfer in a big way. Now, there are some major milestones, in the history of phage, and they relate to its and bacterial genetics, and they relate to our ability to clone, using bacterial genetics. So phage and bacterial genetics continue to this day, and the CRISPR-Cas9 mechanism is an example of how bacterial responses to infection can have interesting molecular mechanisms, and how phage lysis and lysogeny and phage infection can tell us about not just gene regulatory mechanism, but how these mechanisms regulate bacterial life and phage life. Now, this book, again published in multiple editions, can we switch off that light maybe? Is a book by one of the big players in, phage genetics, Mark Potashne. And Mark Potashne looked, let me step back a bit. André Lwoff discovered lysogeny, and he and Francois Jacob and Jacques Monod looked at gene regulation in phage and then in bacteria. And so the whole question about how the lysis-lysogeny switch takes place became a holy grail where everyone ran after, right? And it's just astounding, the beauty of genetics, that the genetic pathway to that was understood by looking at a variety of kinds of mutants which were constitutively active, constitutively inactive, partial loss of function, and combining that with phage recombination and phage infection, the regulatory pathway for phage lambda switching could be uncovered. And then there was a rush to biochemically show, to isolate the molecules involved, particularly the CI repressor, and that again, and its binding was another very important way forward. Now that regulatory loop of what happens between lysis and lysogeny was so captivating that people thought that this is, something which must be applicable to all organisms, and indeed, fundamentally, it is. Unfortunately, let me just see if I misplaced that slide somewhere. Very strange. I'll just move this. Yeah. Yeah, so this interplay between lysis and lysogeny was very, very important. And that's sort of summarized in this, and I won't go into the details, nor should you worry about it. But fundamentally, you have a situation where you have a regulatory loop here between activators and repressors and differential binding to sites of genes, which then can switch between the expression of CRO and CI, right? And this can switch between lysis and lysogeny. And this whole thing can be unraveled by screening for mutants, from which you can infer about these different regulatory regions and these genes. You can have genes which are constitutively active, or sites, binding sites, which are constitutively inactive, and by combinations you can infer all of this. And this is absolutely beautiful to infer how the lytic and lysogenic phase can switch. It gets more and more complicated, and I remember going to lectures on phage regulation. Was it Sudhamai Ghosh who was working on phage in India? Yeah. And so there was a Mahabaleshwar symposium organized by Obaid Siddiqui, and Sudhamai Ghosh was there. And after the lecture, you thought you understood everything, but when you went out, you knew that you had understood nothing. And then, you go back and you read something and you understand it. But this book by Tashneh really explains everything so beautifully. So it's really good to read that. Now, okay, so we learned, and Mark Tashneh, by the way, worked in eukaryotes also afterwards, and the galactose system in yeast was a major area of his work, as well as several others, and how that's regulated, which again is very different, of course, as you know, since it's a eukaryote, but there are very common features. But that links beautifully something very valuable today about how carbon metabolism and gene regulation are linked, right? And today, when we talk a lot about how environment and lifestyle affects our bodies, that's a very important role. And also, we're getting into how metabolism affects, various pathways. And so those pioneering studies are still very important. Okay, so let's move on a bit and keep this in mind, but go on to something slightly different. How did we come to the state where we could understand so beautifully the regulation of genes, and how did we go on from there to understand what happens at the molecular level in your test tube, and how did we reach our current states of understanding? Now, rather than go through that as a sequential history of science, let me switch it a bit and combine that with our own history on this planet. Because in many ways, understanding how we came about and how we are what we are will give us insights into the extraordinary complexities of life on Earth, and therefore the great difficulty in actually in doing something. You might be able to comprehend somewhat, but in terms of doing something to cells, tissue, plants, animals, why is it so horrendously difficult to predict what the outcome of your experiments are. Maybe we can get a hint of that from looking at the history of life on Earth. So the universe came about 13.5 billion years ago. And it's rather extraordinary that we are able to say that with a certain level of precision. And it might be good to look up why we are able to say that. Why not 17? Why not 16? Why not 12? And this comes from the analysis of the current state of the universe, its composition, the speed at which objects and matter and energy is moving away from us, and what the various kinds of predictions are, and going backwards in time. So those are rather precise figures. Another relatively precise figure is that the planet came about close to 4.5 billion years ago, our planet. And life, amazingly, came about perhaps just a billion years after that. So what are the ingredients which allowed life to come about on our planet still remains a mystery, because you can find the fundamental components in many different locations, but somehow we are not able to come to a conclusion about how they came together to give rise to life as we see it. But it appears that life as we know it today has just one origin. It may have had many different origins and failed, but all life today seems to have a common origin, and that common origin came up presumably on Earth, even though people argue that it could have been seeded by something extraterrestrial, but that's all open right now. Now, what we know about, again, this is the beauty of evolutionary biology now, that we can infer that life has a common origin without necessarily getting into molecular biology and understanding all those details. And that comes from the work of Charles Darwin, shown here, along with Alfred Russel Wallace, and they together inferred that all life on Earth, the huge variety, comes about by natural selection over hundreds and hundreds of millions of years, and all life has a common origin, right? Because of the way you can connect life on Earth. And this is extraordinary insight, and really there are not too many new kinds of insights which have come into biology, and there are just maybe one or two more, and this is one such insight. So I'll quickly go through how life came about. About 3.5 billion years ago, simple forms, prokaryotes, came about. A little later, photosynthesis, and that's amazing. You don't have to rely on piped gas or fuel. You can synthesize all the energy you want from the sun. That's amazing. And 2 billion years of complex cells and a billion years of multicellular life. And multicellular life came about by specialization of unicellular organisms cooperating with each other. Then there were, again, amazing changes taking place from 400 to 600 million years. And one dramatic change is the Cambrian explosion about 550 years ago, 570 million years ago, that it looks like the space of gene regulation had created a possibility for the diversification of multicellular organisms into extraordinary forms. How that happened in a relatively fast period, we don't know, but that's a burst of diversification of animal and plant forms of certain kinds, pre-regular plants that we see today. Insects and seeds and land plants all came about in that broad period. Then you had amphibians, reptiles, and 200 million years ago we had mammals coming about. And we had little later birds, but this period of amphibian, reptiles, and flowering plants is very, very important. Dinosaurs dominated the earth more than 100 or 120, 150 million years ago. And flowering plants came about. They resulted in a burst of insects, which I've not shown here, and that pollination resulted in growth of forests of various kinds. And therefore, various kinds of organisms which fed on those forests grew, but yet the mammalian radiation didn't grow substantially. About 65 million years ago, something extraordinary happened. We think an asteroid struck the Earth at that time, at somewhere near Mexico, created huge temperature changes and dust bowls, and for some reason, which people still analyze, seems to have completely eliminated large animals such as dinosaurs. This allowed the mammalian radiation to occupy the space, and the creation of forests by insects and flowering plants working together led to an extraordinary growth in the mammalian radiation. Now, 2.5 million years ago, our ancestors branched out into creating Homo sapiens later on, but Homo neanderthalis, before that Australopithecus, and various kinds of Homo species. And it's only about 200,000 years that we, Homo sapiens, came about. And it appears from the DNA record that we have genes from Neanderthals, Denisovans, and other hominins which coexisted with us. Why we are the only ones who survived? Why others died? We don't know. It appears that their brain and what they could do was by and large very similar to what our brain is and what we can do. And, therefore we don't know what caused their death. We don't know how is it that we mated with them and they with us, and they were sort of very closely related, but they're no longer on this planet. It would have been a very different planet if we had multiple other species like us inhabiting this planet. And the reason why it would have been different is extraordinary. This brain of this Neanderthal and what it can do in terms of making fundamental drawings on caves and so on and so forth is no different substantially. There are differences from our brain. And the earliest Homo sapiens had the ability for language, for art, for culture, for building societies and so on, but they did not do that. They did not do that for tens of thousands of years. What is it that suddenly led to the use of language? What is it that relatively suddenly led to the use of complex and more complex tools? So there are two kinds of metaphors people give for the development of language and the development of culture. One is a jigsaw puzzle metaphor, which says things evolve bit by bit during evolution, taking a very long time. And when those bits come together, new properties emerge. This is the gradualist view and correctly of what most evolutionary changes have taken place. And that's how you see in the fossil record a gradual tendency for new characteristics to come about. And therefore, the argument is there is something which led to the rise of language and communication in the form of language as opposed to relatively trivial forms of communication like what insects do and other animals do. The power for abstraction which we have, for example. So that's the gradualist jigsaw puzzle view. Another metaphor which is used is like the arch in a building which is holding up key components of the building. It's not ready until it's ready. The arch is not ready until it's 100% done. It's not ready when it's 90% done. It's not ready when it's 80% done. It's there and ready only when it's completely done. Right? So that completion somehow came about at some time, and that was an intrinsic property for language, which other people say is the reason why we have language. So there's a gradualist view that components of language were there and there were rudimentary forms of language, The other is that a sudden view that language capability came about. But that was really, whatever it is, that was immensely transformative in the way we work. And the reason for that, which I will not go into it, is because of language and our ability to make tools and form societies, our evolution moved away from mere biological evolution to cultural evolution. The rate of change of cultural evolution is really fast, and our ability for abstraction to understand phage genetics and so on is something which no other animal tries to do. And that's a software solution which has come about, and we don't know yet how this capability came about. So I've talked about how we evolved. Another question which biologists ask three questions. Where did we come from? That's evolution. How are we made? That's developmental biology. And how do we work? That's physiology. The second question is developmental biology. How from a fertilized human egg do we make a human embryo? Right? And there were many theories about this. And again, I will not go into details about that. You can check those out. But fundamentally, the arguments were that there's something in the egg which is partitioned and makes different parts of the organism in the adult. That partitioning can take place by heredity. There's other theories that some influence of the environment of the egg tells the egg what to become. So that's the not what the egg inherits, but where it is, its neighborhood, and variations on these kinds of themes. And this was a field of intense competition and debate, embryology, and different people had very intense views. One of the most beautiful experiments is by Hilde Mangold and Hans Spearman. Hilde Mangold didn't get the Nobel Prize for this work. She died very young in a kitchen in a fire accident. But Hans Spearman got the Nobel Prize, and here is a movie showing Eddie de Robotis at the University of California repeating this experiment. Here are two tadpole embryos. He takes a bit of a tadpole embryo from a place called the dorsal lip and puts it in another location in the other embryo, right? So he'll take this piece out using extremely sophisticated robotics. You can just see it's a pair of forceps and blunt needles, and putting that in another location. And then he goes, so he's taking a piece from here and he's putting it over there. And he goes home and comes back the next day like Spearman and Mangold did, and this embryo has got two axes. So somehow, apparently magically, cells in one location have the capacity to create an axis and put into another location create an ectopic axis. So this is long distance communication. So there were heated debates with various people. On the left is a embryologist, his name is Just, black man who went to Europe and said that in 6 months of Europe he had more collegiality than he had in an entire lifetime in America. And on the right is Goldschmidt, and Goldschmidt moved from America to the University of California at Berkeley, and he said that at 6 months at Berkeley he had more collegiality and was treated well as a Jew than in a lifetime in Europe. And this was a time when there were intense, nationalism and debates over these kinds of things. Anyway, the issue of embryonic development was addressed, and was addressed beautifully when two other fields came together. And those fields were the fields of genetics, understanding how characters are inherited. And it was merely to study a formalism of how characters are inherited. People didn't connect as they did more and more and better genetics, or even as they did phage and bacterial genetics, that this could be used to study the inside of an organism. They studied cow genetics, chicken genetics, dog genetics, and so on, but they didn't know what was happening inside. That insight came by the discovery of the structure of DNA by Crick and Watson using data from Dorothy Hodgkin's lab and Maurice Wilkins' group, and that changed completely. And model organisms became useful to understand not just how external characteristics like the eye and the wing develop and the genetics of that, lots of people worked on that, but how the organism itself was patterned from anterior to posterior, from top to bottom. When recombinant DNA revolution came about, and you could isolate the genes responsible for these mutant phenotypes. And that, again, is a triumph of bacterial genetics, which substantially allowed that. When DNA sequencing came about to scale by Sanger and Gilbert's efforts, where you could actually sequence genes, when RNA in situ hybridization came about so that you could do labeled RNA and look at gene expression, and when you could express genes in bacteria and make proteins and raise antibodies in bunnies, you could look at gene expression pattern inside the animals in different ways, and you could look at homologies between genes by hybridization from species to species. And the amazing discovery, for which several others, Eric Wieschaus, Nusslein Follard, and Ed Lewis got the Nobel Prize, was to show that the toolkit used to construct the fly is identical at a high level with the toolkit used to construct us. So the same genes are expressed in more or less the same patterns at a certain stage in development. And therefore, the analogy there in terms of gene expression is that regulatory genes at a high level are analogous to nuts and bolts and screws and rivets and so on and so forth required to make a complex aircraft. But where to put them, where to connect the sheets of metal, what to do, is the plan which needs to be executed, but the building blocks, the toolkits are the same. So how do you turn on and turn off these regulatory genes and make things is a big challenge. And I'll come to that. So again, the same thing, genes are responsible for making different parts of the animal, people said. And if you remove them, you can make a fly with 4 wings. And the same kinds of mechanisms used to make a fly, the same principles apply to making the human. Now, this led to a view that genes were everything, but there were many studies which tell you how important the environment is. And those came from studies not just in these model organisms like flies or even in humans, for example, but here are ants. All these ants belong to a colony. Their genotype is identical. They become different because they're fed differently, they interact differently in early development, and they grow in very different ways. Here is a worker sitting on top of a queen from the same colony. Right? The queen is huge and eats incessantly. And unlike other organisms such as us, if we eat incessantly, we'll become huge, but we'll die early. The queen is actually the longest lived in the colony. And we know now that it's the insulin pathway which branches in 2 different ways, one which allows longevity and the other which is integrated into metabolism. So there are lots of great insights coming from this. So let me wind down and try to tell you what I think we're learning from this, which is perhaps will be useful for us. We're made by these kinds of approaches and continue to make discovery upon discovery about how things work. But there's nothing which we have found which is fundamentally new. In other words, there is no new law of biology which we have discovered. All of biology is consistent with what we know are the rules of chemistry and the laws of physics, right? So there's no magic or something new which people used to speculate about, that the organized life requires a new law. Why do they think that organized life requires a new law? The argument made by physicists was living organisms have order. Unless there's a way by which you preserve that order, you will result in the organism rapidly dying and having disorder, or the organism not even being made in any way. In other words, low entropy is required for life, but high entropy is the natural ground state. And unless you keep pumping in energy, you can't have, a low entropy. And therefore, the view about life is that for having order inside, you need to have disorder outside because you're pumping in energy from outside into inside, and you can have a system which is one way open where energy comes in, but you keep the order inside. Now, separate from this work in biology, there were others looking at how algorithms, as we call them today, how computing works. And there was a person, Alan Turing, who was working in Cambridge and then Oxford, and then running between Cambridge and Oxford during the Second World War, trying to decode the Nazi code at Bletchley. And Turing was a famous, made enormous contributions I'll come to later. But Turing created a machine mentally, the Turing computer, which, given enough memory, could compute anything, right? So it's a set of instructions on how to compute anything. You can just go on, and therefore this was the stored program computer in theory, and there are many of the computers now are versions of that stored program computer. There was another scientist, John von Neumann. Again, his work was not recognized by biologists, who said, let's try to replicate the stored program computer. And von Neumann said, if you want to have the instructions to replicate it in the computer itself, you cannot do that unless you have an outside system orchestrating this process. In other words, to have the computer replicating itself, have the instructions to replicate itself, and to compute, he said was not feasible. And he also said that when you replicate, this controller which replicates it makes mistakes, and therefore the daughter computer which you get will be a mutant form of the mother computer, and so on and so forth. So mutations will happen, and therefore the computer will change, right? And this was great insight, and this work went unnoticed by biologists. But this was a very biological statement to make. Now, during the Second World War, this man, Schrödinger, who was a genius, but news coming out points out that he wasn't a very nice man. Schrödinger was in Dublin in exile, and Schrödinger was fascinated by an experiment or a calculation which Max Delbrück and Nikolai Timofeev-Rassovsky did. And what Delbrück and Timofeev-Rassovsky did was to look at the genetics, the work of Muller. And Hermann Muller used X-rays to make mutant flies and showed that X-ray was a mutagen. And what Delbrück and Timofeev-Rassovsky reasoned was, if the DNA is a target, right, or the genetic material is the target, and you're shooting an X-ray like the man there is shooting the camera at me, you're shooting the X-ray at the target, then the frequency of mutations in specific genes will give you an estimate of the size of that gene. If a gene is very big, you'll get high frequency mutation. If a gene is very small, you'll get low frequency of mutations, right? And so Delbrück and Timofeev-Rassovsky did a calculation saying, if that's the target, and that's the source, and that's the energy of the source, let's calculate the size of a gene. And they came up with a number. And some years later, when Delbrück was asked to comment on this work, he said, it's complete rubbish. There are too many variables, but there's too much guesswork going on. Delbrück also, by the way, when asked why he got the Nobel Prize, he said he got it for hanging on long enough. Many of his students had already got the Nobel Prize. It was embarrassing not to give it to him. Okay, so Schrödinger took that information from that paper, and that was one of the key aspects of his book, What is Life?, in which he talked about the way life comes about, all this issue about thermodynamics and so on, and he talked about the nature of the gene. And he said something, that to make this embryo, not this embryo, but to make an embryo from an egg, a fertilized egg, there are rules which are laid down, and those rules are laid down in the DNA. And he used a phrase, amongst other phrases, that the genetic material, the chromosome, as they thought at that time from Muller's and Morgan's work, was architect's plan and builder's craft. In other words, you had a plan on how to make this building, but the executor of that plan was also in the plan itself. You don't need your construction agency to come in and your masonry to look at, you have to put this there and so on. Everything is there, right? That it is both put there together. So in other words, to him, genes were everything, and there was a way by which this auto-regulation, you make regulatory components, you set things up and you get it done. So that was Schrodinger's view. Now Turing, as I said, did enormous work. He worked on patterns in development, the Turing machine. He also worked on something which is very relevant today, which is the Turing test. And the Turing test asks, how do I know if any of you is a computer and which can mimic humans, right? And therefore, if the computer passes the Turing test, in which I cannot distinguish it when it asks a question, when it answers a question from a human, then it passes the Turing test. And that's being used a lot, and much of our artificial intelligence today passes many, many Turing tests. But coming back to Schrodinger, Schrodinger said, and he made this mistake, on the Turing stored program computer, and I told you that von Neumann showed that the description is separate from the constructor. Schrodinger said that chromosomes are architect's plan and builder's craft in one. The code script actually just contains a description of the executive function. It is not the executive function. So the fundamental question then comes up, what is the executive function? And in many ways, that was staring at us from the earliest days of phage and bacterial genetics. The executive function is not in the DNA sequence, even though the DNA sequence encodes your repressor and your activator and so on and so forth, right? All of that is there. But the executive function lies elsewhere, and according to Sidney Brenner, to understand the constructor part of the machinery, the cell is the right level of abstraction. Cell products which are there, cells interacting with the environment, are the executive function. So, to look at this again, this cellular milieu is what changes the regulatory setup. And in today's world, where we can have enormous amount of data, on the top left is the Waddington's landscape, showing how, starting from the embryo, cells can make different kinds of choices going down a landscape. Below that, all these threads are, pulls and pushes of DNA expression, which shape the landscape, but they're not the landscape itself. And you can look at gene expression patterns at receptors and activators and repressors and see how something rolls down the landscape. And you can do this with big data, you can do it with many genes at the same time, and therefore computational biology of this kind is taking it out of this tsunami of big data which we didn't know how to understand in a multidimensional way, and therefore our best approach to understanding modern developmental biology or gene expression and its consequences are collaborating with experts who do various theories like this. So I'll end here, even though there's lots of other stuff to go. It's not that mathematics can solve biology or computational science can solve biology. It's not that we don't have, like in physics, if you understand the laws of physics, you can predict planetary motion precisely. You can't do that with biology. Biology just about manages and we get along. In this situation, understanding something as complicated as the brain, as distinct from a bacteriophage, where there are 86 billion neurons and tens of thousands of genes, but huge numbers of base pairs of regulatory information which we don't know, how gene regulation results in brain development and movement is a very exciting area. And as long as we keep in mind that understanding biology is understanding algorithms and not understanding laws, and deciphering those algorithms in different contexts is our task, I think we have great exciting times ahead. Thank you.
You can be seated now. Thanks. Thanks, Vijay, for the fascinating talk. I think we all went through a beautiful journey from phages to human evolution that reminded me of a quote that nothing in biology makes sense except in the light of evolution. That was also part of it, and thank you so much. And I request BG to hand over a small memento to Vijay.
Thank you very much.
So we have refreshments. We have refreshments arranged in the canteen. Please move on there, and you can help yourselves.
Yeah.
Yeah. Okay.
I'll just disconnect this and join you.
Sure.